we go for the next presentation. And I will give for Dr. Naveen to present Professor Jürgen.
So it is my great pleasure to introduce Dr. Jürgen Rabenhorst to the August gathering here. We all know about Dr. Rabenhorst. He is a very renowned scientist, and he is a professor of industrial biotechnology in Technische Hochschule in Ostwestfalen, Lippe University of Applied Sciences and Arts in Lemgo, Germany. And with this brief introduction, I'll request Dr. Rabenhorst to enlighten us with his knowledge and give his presentation. I invite Dr. Rabenhorst to present his lecture. Thank you, sir.
Okay, thank you. It's always better if I happen to introduce myself. And I just want to mention before I give the lecture, which is already recorded, but this is a part of the German-Malaysian project, which is funded by the BMBF. And the work which I present is mainly done by a postdoc of mine who unfortunately has left the group a couple of weeks ago, but she already made the presentation, and I would like to ask that the presentation is now displayed.
All right. Dr. Jürgen, you sent me your recording, so I will play it right now.
Okay, thank you.
Dr. Jürgen. Okay, just give me a moment. All right. All right, let's share the screen. There it is. Okay, can everyone see?
Yes.
All right, the following presentation is Dr. Jürgen's presentation, so let me know if there are any problems with the volume. I'll play right now.
Thank you, Isan.
Welcome to my lecture, Enzymatic Digestion, leading to cultivation medium product of Cellulose plantarum. This presentation.
A little bit louder, please.
Okay.
Isam, could you increase the sound more? 100.
Oil palm sugar, which is done in the Department of Life Science Technologies at the University of Applied Sciences and Arts in Lemgo. And what I'm presenting today is mostly done by Pia Neubauer, who is a postdoc in my lab, and in part by Rabea Dirkes, who is also working in this project and will give a lecture directly after my lecture. The project Oil Palm Sugar is an abbreviation for the project name Synergistic Use of Oil Palm Wood for the production of pressed composite materials and the utilization of the remaining waste for the sustainable production of chemicals by fermentation of carbohydrates. The objective of his projects were different departments are working together synergistically, and the departments who are working for is the Department of Life Science Technology in Lemgo, where also my lab is located, of industrial biotechnology. It is the wood technology at our university, and we have a couple of partners, an industrial partner here in Germany, which is LED Enzymes, who delivered us with the enzymes, and in Malaysia, FIDIC and FRIM, who are working together with the Department of Wood Technology and the University of Technology Malaysia, they are the Institute of Bioproduct Development, who are cooperating with us. The topic where Pia Neubauer worked on is the enzymatic hydrolysis of palm trunks to a feasible cultivation medium for the fermentation to industrial relevant chemicals. Here you can see an overview of what we are doing. We use the oil palm trunks, and in this case, the sawdust from oil palm trunks. We do a pre-treatment with that material before we go to the enzymatic hydrolysis. And at the end, we will cultivate the obtained sugar solution with Lactobacillus plantarum as a typical example for bacterial cultivation, because Lactobacillus plantarum is a producer of lactic acid and also a probiotic bacteria. Here you see the workflow, how we worked on that. We started with sawdust from the OPT, which we obtained from the wood technology department. And this was followed by a pre-treatment, which was done at 122 degrees for 20 minutes in an autoclave, where we added either sodium hydroxide or sulfuric acid. And followed by that, we have an enzymatic hydrolysis reaction with cellulases and xylanases leading to a glucose solution. And we tested here two alternatives: the washing of a pre-treated material or use of a material without any washing. And at the end, we had the cultivation of the obtained hydrolysate with Lactobacillus plantarum to see whether this process works. And you can see here in the next chart the results of the enzymatic hydrolysis after the pre-treatment of a wood. On the left-hand side, so you see the results with washing, and on the right-hand side without washing. And the gray bars you see here are the material which was treated with sodium hydroxide, while the red bars are the samples which were treated with sulfuric acid. And as you can see, for the washed samples, the sodium hydroxide is much better. We obtained glucose concentrations of up to 537 grams per kg dry material. And with washing, we only have very low concentrations when we have treated that before with sulfuric acid. And without washing, you see there are the difference between the both treatment methods not that large, but in both cases, the concentration of the sugars are much lower. But in this case, we had better results for sulfuric acid treatment. What we can say as a result of that, that the unwashed materials, we only were able to hydrolyze 52% of the cellulose by higher sodium hydroxide concentrations, while in the case of washing, we obtain a hydrolysis of 67% of cellulose with 10% of sodium hydroxide. And when we use these obtained hydrolysates directly for the cultivation of Lactobacillus plantarum, we have here the results of a material to which the washing step was applied, and on the right-hand side without washing. And you can see here the proportion of either the base or the acid. And you can see with increasing concentration of a base here in the upper part, we have a slightly increasing concentration in lactic acid, but the remaining glucose, as we have a higher glucose concentration here with a higher concentrate, is much higher here. So in general, we can say that the obtained hydrolysate, which was not washed and even which was prepared with a high concentration of sodium hydroxide, we have a good production, even higher production compared to the low concentrations, but that might also depend that here the glucose concentrations might begin after 24 hours to be limited. Without washing, you can see we have lower glucose concentrations and even not that high concentrations of lactic acid, but also we see an increase with the higher concentrations of up to 8.5 grams per liter lactic acid. And for sulfuric, we can see as we have a very low glucose yield obtained with washing. Also, the concentration of lactic acid is very low. Without washing, it looks a little bit better, but Without washing, it looks a little bit better, but still the values are lower compared to sodium hydroxide. As it was mentioned in literature that due to the pretreatment by high temperatures and extreme pH values, there might be the formation of inhibitors, we added 2.5% charcoal to the obtained hydrolyzed and incubated it by shaking for 5 hours, and then use these materials and compare it with the unwashed material. And you can see here the results again after 24 hours. And again, we see that with an increase of the base, sodium hydroxide, we have an increase in lactic acid formation and a higher concentration in glucose. And when we incubate it with charcoal, it looks like that it might be even at the higher concentration of 3.5 and 5% of sodium hydroxide better than the unwashed material. And for sulfuric acid, again, we see here also an increase, and the amounts here seems to be comparable, at least not better at the lower concentration. It could be that even the charcoal may reduce the lactic acid formation. So we can conclude that the incubation with charcoal did not really improve the lactic acid yield, so that we can leave out that step. As we have seen that the lactic acid production with the hydrolyzed solution is in the yield comparable to the existing MRS medium. So it's now important to have a look at the cost calculation to get an impression of the economic feasibility. Despite the fact that wood is a waste product, the pretreatment and also the enzymes are the cost-pushing steps. The MRS medium costs about 30 eurocents per liter. The sugary solution, which results from the washed pretreatment with 10% sodium hydroxide and the following enzymatic hydrolysis costs are in the range of about 50 eurocents per liter, calculated for costs in the lab scale in Germany. The main cost driver is the price of the enzyme solution. By decreasing the enzyme costs down to 10 euro per kg, the price of an MRS medium can either be met or just even undercut, depending on the yields of the glucose, which is obtained out of the hydrolyses. So this shows that it is economical feasible to use this waste material, the soft parts of oil palm wood, as a raw material for enzymatic hydrolysis, for the utilization as a medium for biotechnological processes. So coming to the conclusions, we were able to show that heat treatment with sodium hydroxide the wood hydrolysis with a highly active enzyme mixture was successful. It is important to have enzymes which are really active on the hydrolysis of a cellulosic material so that we can obtain a high amount of glucose from the cellulose. The highest glucose yields we achieved after the pretreatment with a 10% sodium hydroxide solution and following washing steps, which resulted in a glucose concentration of 34 grams per liter and a 65% yield of glucose out of a cellulose. Without washing, the process would be cheaper and more sustainable, but the yields of glucose were 5 times lower. So from an economical point of view, this is not an alternative, or we need still further optimization to avoid the washing step. We have shown that the subsequent cultivation with Lactobacillus plantarum with a pH adjusted and sterilized solution yielded in up to 11 grams per liter of lactic acid within 24 hours. This is comparable to the concentrations we obtained with the typical standard media which are used for lactic acid bacteria. In addition, the obtained biomass out of this process of Lactobacillus plantarum is also of interest because Lactobacillus plantarum is a well-known probiotic bacterium which can be used, for example, for feed for animals. And we were able to show that with decreased enzyme costs at an industrial scale of about 10 euros per kg, the process is competitive to the lactic acid production with standard media. This is important because the raw material costs of the media are an important factor in the production costs of these bulk chemicals. And the cost estimation revealed that the enzyme costs are the crucial point within this process. But as we got the information from our industrial partner as well as out of literature, it is possible to obtain enzyme costs in the range of 10 euro per kg for cellulases if it is used in an industrial scale. And at the end, I would like to thank my co-workers, Pia Neubauer and Rabea Dirkes, who did the work. I would like to thank the colleagues from Biotechnology who supplied us with the sawdust of the wood, the German BMBF for funding, and AB Enzymes for delivering the enzymes. And I thank you for your attention, and I'm ready to answer your questions.